The first type of tissue that we're going to talk about in this chapter is epithelium. So, one of the characteristics of epithelium is that it's responsible for the covering of surfaces and forming glands as well. These cells have what are called polarity. Remember, a water molecule is a polar covalent molecule because it has two ends to it. So in this case, the cell is going to have two ends. It's going to have an apical end and a basal lamina end. So if we look at this word basal lamina, it kind of looks like basement. So when we think of a simple columnar cell, the top of it is going to be the apical end. It's what's going to face whatever is traveling within the respiratory system or digestive system. It's also going to have a basal layer, which is going to be facing the connective tissue. Another characteristic of epithelium is that the cells are closely packed together. So when you differentiate between different types of tissues, this is one important characteristic that's only in epithelium. With connective tissue, the cells have a lot of space between them. It's supported by connective tissue, reticular lamina, which is under the basement lamina. So if we have our example of a simple columnar cell, there's going to be connective tissue, which is under that. So this blue structure would be connective tissue, and this would be the epithelial cell. So if you were to get a deep cut and it would go down through your epithelium, deep to that, you would see the connective tissue. A very important characteristic of epithelium is that it's avascular and innervated. Avascular means no blood supply. So the first place that we find blood supply is in the connective tissue. Innervated means that there are nerve receptors. Recall that on the last slide for sensory reception, I mentioned a type of stratified squamous epithelium. It has a high rate of regeneration. And this is important, especially for areas like the tongue or the mouth, the oral cavity, where there could be damage to superficial cells. Those deeper cells need to regenerate to replace those more superficial cells. And finally, it can be one layer, which means it's simple, or it can be many layers, which means it's stratified. So when you name epithelial tissue, there's two criteria you need to use. You need to use a term for the number of cell layers here and also for cell shape. So for cell layers, you're either going to say simple or stratified. And for the shape, you're either going to say squamous, cuboidal, or columnar. So we would have things like simple squamous epithelium or simple cuboidal epithelium, simple columnar epithelium, or we could have stratified squamous epithelium. And there are stratified cuboidal and columnar, but you're not going to be responsible for those. So you're going to kind of, it's not going to be one of the slides, and you're going to kind of skip through that part of the text. So let's begin by looking at simple squamous epithelium. And simple squamous epithelium is a single layer of flattened cells, usually as a central nucleus, and it's the simplest of the epithelium. So you need to know the description, function, and location of each of these. And again, it's a really good idea to put these on index cards to study from. And its function is that it allows for materials to pass via diffusion and filtration. And there's also secreting lubricating substances in serosa. which is something you learned about in chapter 1. So, for example, there was things like the visceral pericardium. That would be the layer that is directly touching the heart. And then there is a parietal pericardium. These would be two examples of serosa. These specific membranes are going to be producing liquid. So the location would be in places like the kidney glomeruli, air sacs of the lungs, lining of the heart, blood vessels, lymphatic vessels, and serosa, like the visceral and pericardial layers. Now, what we're seeing on this image right here is from the lungs. So this is an air sac of the lung tissue. It happens to be called what is an alveolus, and it's where the gas exchange is going to happen. The beauty of these squamous epithelial cells is they don't require a really thick boundary. So it's super easy for oxygen and carbon dioxide to diffuse across the membrane. And once they diffuse, they go directly into the pulmonary capillaries and then travel back to the left side of the heart where they're going to then go to the entire body. The next one is another simple layer, but the shape is somewhat different. They are cube shaped. So they are a single layer of cube-like cells, and you can tell a lot by the shape of the nucleus. In this case, the nucleus is kind of spherical and it's central. And the nucleus is going to fill up much of the cell. So I would encourage you for your lab not only to look at the slides that your instructor shows you, but also to use Practice Anatomy Lab, which is found in your Pearson Mastering A&P, because it has lots of various images. When you learn anatomy, the best thing to do is to learn to see all the various examples so you can compare and contrast it so that hopefully certain things will stick out. Its function is going to be secretion and absorption. I like to think of these cells acting like a sponge. So a sponge can absorb dirt and water that's on your counter, but you can also wring it out, and that would be secretion. So the location we find this is in the kidney tubules, ducts, and secretory portions of small glands, as well as the ovary surface. So you can see here that these are the simple cuboidal epithelial cells. There's basically one nucleus per cell. And in this case, this is what's called the kidney tubules, which you don't need to know what those are now. You'll learn more about them next semester. But what they are, it's really interesting. They are tubes that descend down into a part of the kidney called the medulla. And those cells are going to be the site for absorption of molecules and for the secretion of molecules. When there's absorption of molecules, they go into the blood stream.